I'll be talking about this funny phenomenon called Ephaptic coupling in the context of EEG and white matter. So let's explore. So EEG has been around for roughly 100 years now, human EEG. And since this time, we've mapped lots of different signatures to different behavioral processes, sometimes through just signal processing or AI decoding, etc. But unfortunately, when you do brain mapping classically, you end up testing something under a controlled condition. And this doesn't really reveal mechanism. It often leads to these paradigm-bound theories. So what would be much more interesting is if we could have a generative model built with biologically plausible subcomponents that can recapitulate EEG data and also perform tasks and predict human behaviors. So, in the context of EEG, an attractive generative model would be able to explain a whole lot of different phenomena: cross-frequency phase amplitude couplings, individual alpha peak differences, these funny traveling cortical waves that you can see, and of course, the scale-free log linear 1/f, which I've always been fascinated by. Because many aspects of nature, such as the sounds of oceans, even Saturn's rings, are actually follow this 1/f log linear pattern. And of course, we see it in EEG and MEG signals, as well as ion channel gating. So what are we actually measuring with EEG? This is kind of the classical model of what we're actually measuring. So in theory, what we're measuring are these radial dipoles that arise due to something like 10 to the 6th inputs into dendrites, the apical dendrites, mostly from pyramidal cells in layer 5. So 10 to the 6th neurons have to fire quasi-synchronously within roughly 2 centimeters in order to produce a signal large enough to propagate to the scalp. And generally, axonal conduction is considered negligible because it's thought to be too asynchronous to summate. But what if white matter did contribute to EEG signals? How could that be possible? So there's a few clues here that I think are interesting. So first of all, when we're born, we have something like double the neurons that we're going to have by age 2. There's just massive amounts of apoptosis during that early period of development. But another thing that happens is that the white matter tracks begin to be myelinated. And what parallels this myelination is changes in the slope of the 1/f, as well as the emergence of the alpha rhythm. So infants don't have an alpha peak that you see very clearly in adults. And another thing that I think is very interesting, I did not know this, but white matter tracks have unmyelinated neurons. So for many years, the fixative technique used in electron microscopy would preferentially kill off the unmyelinated neurons. So even in large tracts like the corpus callosum, in humans, about a third of the neurons are actually unmyelinated. So what are they doing? Because you wouldn't expect the signal to be able to travel that far in an unmyelinated neuron. But one of the consequences of these unmyelinated neurons is that it increases the fiber packing density, and thus it increases the transverse resistivity of the local environment within the white matter microstructure. So when the resistivity is increased, it helps promote this really interesting phenomenon called ephaptic coupling. So ephaptic coupling was first observed in the 40s when some scientists put 2 different axons parallel to each other in a very highly resistive medium, something like paraffin, and they found that one signal traveling down one axon could actually cause an action potential in the nearby neuron. But that was artificially high resistivity, not biologically plausible. What I expect is that we might have a much more subtle effect in white matter. So when the effect is more subtle, what can happen is that action potentials that are initially the initial onset has a slight phase delay, they can actually resynchronize due to this ephaptic coupling effect. At extremely high ephaptic coupling, you can actually see a desynchronization. So there's kind of this interesting phenomenon that can actually promote more synchronization in the action potentials firing and traveling along the white matter tracks. And there's a variety of different mechanisms how this could happen. Another one to think about would be this mechanosensitivity, because we have mechanosensitive voltage-gated channels all along the axon. So I created this model using cable theory. Sorry, I'm running out of time. Human connectome project data, white matter as input, and you put it through this model, and voila, you get these spectral densities that look very realistic. You can estimate the ephaptic coupling strength. And then I wanted to validate this even further. So I used something called transcranial ultrasound to suppress activity in the corpus callosum specifically. And it turns out that this is baseline sham, and the condition that you see alpha desynchronization when you apply TUS to the corpus callosum. And interestingly, you don't see it in the main areas that were putatively thought to be alpha generators. So I think that's, my time has probably run out, but I think that this is a very interesting phenomenon that deserves, further study. And I think that possibly dynamic changes in white matter may subtend certain things like learning and memory. So, yay. Thank you all so much.
Wow. Okay. Questions,
What are some of the mechanisms that might tune the degrees of ephaptic coupling to get in the synchronized regime, or like tugging too hard and desyncing? Like, are the glia involved, or?
Yeah, I think so. I think that the extent to which the nodes of Rambier are aligned may affect the strength of ephaptic coupling. I also think, just the amount of myelin as well as the ratio of myelinated to unmyelinated neurons. But yeah, that's a great question. I think there's a lot to explore there. Yeah. You didn't ask one yet?
I'm going to do a slightly annoying thing
Okay.
and point you to one of my papers. I'm very sorry, but so I worked on a project where we did light microscopy and reconstructed single neurons in the mouse cortex, and there we found this very weird thing that we saw neurons in the motor cortex that made these very long axons and went all the way to the corpus callosum, and then entered on the contralateral side and then just stopped and did nothing, and it was very consistent between samples. So, is it so? Do you think some of these neurons could actually, even if they just run through there, they can have an influence on the synchronicity of the connections? That is wild.
Yes.
Okay, that's really cool.
Because I think the brain doesn't keep, an appendix around.
Right.
It must have
right.
some purpose. So I think
Exactly.
just increasing the resistivity and promoting this synchronization might be the purpose.
It's also
for it.
so metabolically costly to maintain these things.
Exactly.
Yeah. Wow.
Yes.
That's really cool. Okay,
Yes.
thanks.
Yeah. Thank you guys so much.
so much. This was great.